A-Level Biology 9700, Paper 5, Variant 1, May-June 2025, question number 1. So question number 1 is, pepsin is an enzyme that is present in gastric juice. Gastric juice is secreted into the stomach of humans and many other animals. Pepsin catalyzes the hydrolysis of proteins into peptides as shown in figure 1.1. A large molecule of protein has been broken down into peptides with the help of this pepsin. So egg albumin, egg white, contains a high proportion of protein. 10% albumin solution is a cloudy white color. This becomes colorless when pepsin is added to the 10% albumin solution. Our student used a colorimeter to follow the progress of the hydrolysis of protein by pepsin. The student used a blue filter in the colorimeter. Outline one other step the student should carry out to prepare the colorimeter so that the correct measurement of absorbance can be obtained. So, well, this question is about the preparation of the colorimeter. So, how we can prepare a colorimeter? Because the most critical step is to prepare a colorimeter. That is, it's to calibrate it or zeroing the device. That is, the colorimeter should be on the zero. So to do this, the student should use a blank to zero the colorimeter, or the student should place a cuvette filled with distilled water, that is, and then press the button to set the absorbance to zero. So in this way, this ensures that any absorbance measured during the experiment is due only due to the changing cloudiness of the albumin, not the plastic of the cuvette or the solvent itself. So this point can be written as an answer. That is, first of all, I would use a blank. Student should use a blank. To zero the colorimeter. Oh, it filled with distilled water. Press the button to set the absorbance. So, moving towards the next part. After preparing the chlorimeter, the student made a pH2 albumin solution by mixing 10 cubic centimeter of 10% albumin solution with 10 cubic centimeter of pH2 buffer solution. So added 3 cubic centimeter of the pH2 albumin solution to a chlorimeter tube, added a small volume of Pepsin solution to the chlorimeter tube, immediately placed the chlorimeter tube in the chlorimeter. So recorded the absorbance of the mixture every 20 seconds per 5 minutes, calculated the rate of change in absorbance as a measure of the rate of the protein hydrolysis. The results are shown in figure 1.2. So we are going to move towards the next part of this question. So you can see a graph has been shown in this figure 1.2, and on one side of the graph time has been shown and the other absorbance in EU units. So this graph shows the different values of the different levels of absorbance in different time measurements. So use figure 1.2 to calculate the rate of change in absorbance or at pH2 between 40 seconds and 120 seconds. Show your working and include appropriate units. Well, we will move first of all towards the graph to find out the values between 40 second and 120 second. So we will go, yes. So at 40 seconds, you can see the value can be, this is the 40 seconds. Do it with the help of the scale. I cannot do with scale because I am drawing on my laptop screen. And the other value, which is 120 seconds. So at 120 seconds, and there should be a single line as well, not just like me, because so the values are this value and this value. So this value is 0.52. It is above 0.52 and it is 1, 2, 3, about 0.23. So then we will calculate. So for calculation, first of all, we will identify that what are the coordinates. So at 40 degrees, the absorbance is 0.52 and at 120, it is 0.23. So at 40 seconds, the absorbance is 0.52 AU and at 120 seconds, it is 0.238. And the calculation formula is rate is equal to change in absorbance divided by change in time. Change in time, which is equal to, we can write in this way, y2 minus y1 on y-axis and the other on the x2 minus x1. So we will find out this. First of all, we will find out the absorbance, which is in this way. Change in absorbance. Is 0.23. I think we should write over here. Mm, it is equal to 0.23 minus 0.52. It is equal to minus 0.29. We have to write the units as well. Now the change in time. 120, 40 seconds. So it is equal to 80 seconds. So the rate is 0.29 minus 0.29 divided by 80. So its answer is equal to minus 0.00036 AU minus 1. So this is the answer. Now we will move towards the next question. And next part is B. The student decided to investigate the effect of pH on the rate of protein hydrolysis by pepsin. From the internet, the student found out that pepsin is inactive at pH 6.5 and above. The student was provided with a calorimeter and standard laboratory apparatus. Describe a method that the student would use to investigate the effect of pH on the rate of protein hydrolysis by pepsin and to determine the optimum pH of pepsin. So your method should be set out in logical order and be detailed enough to allow another person to follow it. So after calculating values, now we have to investigate the effect of pH on pepsin activity and determine its optimum pH. So for this purpose, you can see we have to uh, keep different factors. So first of all, the key principle is to keep all factors constant as, for example, the temperature, enzyme concentration, substrate concentration. So while we are using the different values of pH, so for this purpose, we have to prepare a range of pH solutions. For this, we have to set up the LAS, at least 5 different test tubes. So each containing a buffer solution at a specific pH. And then we have to add the substrate into each substrate at a fixed volume. That is the 10% albumin solution. Then we had to equilibrate temperature to place all test tubes in a warm uh, in a water bath set to a constant temperature. As for example, the temperature should be 37 degrees or 40 degrees centigrade for 5 minutes to ensure that reach the same temperature before starting the reaction. Then we have to add the enzymes or add a fixed small volume of pepsin solution to the first tube and start a stopwatch immediately. Then measure the rate, transfer the mixture to a cuvette and place it in the calorimeter. So record the absorbance at regular intervals, then for every 20 seconds for a fixed duration. Alternatively, we can measure the time taken for the cloudy solution to become clear, then repeat or follow the same procedure for the remaining pH buffer tubes, ensuring the volume of albumin and pepsin remain identical. Then we can analyze the data by calculate the initial rate of hydrolysis for each pH by determining the change in absorbance over time, or we can plot a graph, we can make a table, so determining the optimum pH, the optimum pH is the point, or your graph where the rate of reaction is at its maximum. So in this way, we can write its answer in detail, which I have told you. So we will start the detail of this question as in this way. So while we have specific place to write the answer, so I will start from the preparation. So prepare a range of pH solution about at least 5 different test tubes. Each containing a solution, buffer solution at specific pH. To each tube, add a fixed volume of the 10% 10% albumin solution. Place all test tubes in a water bath. In a water bath, set to a constant temperature. that is 35 to 40 degrees centigrade. For 5 for 5 minutes to ensure they reach the same temperature before starting the reaction. Note a fixed small volume of Pepsin solution to the first tube and start a stopwatch. Immediately. Transfer the mixture to a goblet and place it in the dosimeter. Record the absorbance at regular intervals. For a fixed duration. Alternatively, measure the time as well. For hydrolysis, repeat the and repeat the same procedure for all tubes. make calculations, plot graph between rate of reaction and pH. So it was all about this question. Now move towards the next question, and next question is complete the sketch graph in figure 1.3 to predict the effect of pH on the rate of pepsin hydrolysis by pepsin. So on one side they have given us pH, and on the other side we have to take the rate of protein hydrolysis. Rate of protein hydrolysis. The maximum should be at between 2 to 2.5. So the graph will be go in this way. So why maximum 2 to 2.5? You know, the pepsin work best. Pepsin work best in acidic condition. And the pH of stomach solution or the gastric solution is between 2. So graph will be just like this. This has been also given in marking scheme. Now the next part is C, which is laryngopharyngeal reflux, LPR, is a disease that occurs when gastric juice from the stomach moves up to the esophagus and into the larynx. So larynx is a voice box which has a laryngeal epithelium membrane, and trachea, esophagus, and the movement of gastric juice has been shown in this figure 1.4. So gastric juice can damage the laryngeal epithelium. Gastric juice contains hydrochloric acid and pepsin. Some scientists identify a reduction in the quantity of two proteins, CA3 and CEP70, present in the laryngeal epithelium of people with LPR. The scientists obtained 6 sections of mammalian laryngeal epithelium to investigate how different test conditions affect the quantity of CA3 and SCP70 in the laryngeal epithelium. Each section of the laryngeal epithelium was exposed to different test conditions for 20 minutes. Pepsin is inactive in the presence of the inhibitor peptide. So pepstatin is, here you can see the different test conditions 1, 2, 3, 4, 5, 6, and pepstatin inhibitor. In the first two conditions, it was not present, and the other it was present. So the pH buffer was 7.4 and 4, then 7.4 and 4. So alternatively, it was 7.4 and 4. So proteins were the extracted from the sections of the laryngeal epithelium. The proteins were separated using electrophoresis, CA3 and SCP-70 were identified using fluorescent antibodies. So shows the results of the fluorine. So SEP70 and SA3 is lower and the wells are there. Electrophoresis gel has been used. You know the process of electrophoresis. So use table 1.1 and figure 1.5 to state the conclusions that can be made from the results of the protein electrophoresis. So here you can see the condition 5 and 6. They have negative control. That is the pepsin. Negative control, no pepsin was added into them. So the bands for SCP-70 and CA3 are thick and dark at bottom, pH 7.4, 7.4 and pH 4 in 5 and 6. So condition 1, you can see there is pepsin is present at pH 7.4. The band remained dark. This shows pepsin is inactive at pH 7.4. The effect of pepsin condition 2, you can see it is condition 2 pepsin is present at pH 4. The bands for SCP-70 are extremely faint. So we can conclude that pepsin is active at pH 4 and digests breakdowns both SCP-70 and CA3 proteins. You can see this condition. The band is thin or almost invisible. And other conclusion we can make that condition pepsin is present, but peps is also added. So pepstatin is also added here. So the bands for N are dark and thick again. So peps, this is the enzyme inhibitor. So successfully inhibits pepsin, preventing it from digesting the laryngeal proteins even at its optimal pH. So we will, with this we can make a conclusion that is the pepsin requires an acidic environment pH 4 to digest SCP-70 and SCA3. So it is inactive at a neutral pH. So these are the 2 points which should we write. I have explained you in detail. Now I would write in briefly. Pepsin requires an acidic environment. pH 4 is suitable over here to digest SCP-70 and CA3 proteins. It is inactive at neutral pH 7.4. And second is pepstatin is an effective inhibitor of pepsin, protecting these proteins from degradation. So you can see here it is, whereas moving towards the next question. The results of this investigation were published in the scientific paper. A student who read the scientific paper concluded that pepsin causes damage to the laryngeal epithelium in people with LPR, suggests why the results of this investigation might not support this conclusion. So, as we know that in the if we discuss previous data, uh, here is an explanation of why the investigation results might not fully support the conclusion that pepsin causes damage to the laryngeal epithelium in people with laryngopharyngeal reflux. In scientific practicals like this, we you can see uh, we need a laboratory experiment and a real life medical condition in vivo. So here in this case, the conditions were different. The experiment was conducted in a laboratory using extracted protein and buffers. In a living person, the larynx has protective mechanism like mucus and constant cell renewal that are not present in a test tube. So this can be one reason. And the second is the experiment uses fixed pH buffers, that is 7.4 and 4 in a person with LPR, and the pH in the throat fluctuates constantly and may not stay at pH 4 long enough for significant protein digestion to look. And then the the electrophoresis results only show that two specific proteins and are broken down. This does not necessarily prove that the entire laryngeal epithelium is damaged or that function is lost. So we can say that the study was done in vitro. So results may not reflect what happens in a living body. So protein degradation, which is not the same as clinical tissues damage or disease symptoms. So I am going to write over these two points. The study was done in lab outside the living organism. So results may not reflect what happens in a living human body. The second is the study only shows protein degradation which is not the same which is not the same as clinical tissue damage. Yes, the human body is different. Well, it was all about the question number one. So please like, share, and subscribe my channel. And thanks for watching.